All right, hello everyone. Welcome back to the SNRE Graduate Seminar. We are excited to have you here today and welcome everyone who is joining us on our live YouTube stream. Today we are going to have a presentation from Dr. Lily Kirk and Dr. Matt Cohen will be introducing her. So go ahead, Matt.
Yeah, hi everybody. So yeah, it's my very great pleasure to introduce Dr. Lily Kirk to you for this afternoon's seminar. Lily was a PhD student here at the University of Florida until just a couple months ago when she successfully defended her dissertation and got through the graduate school. So she's now officially a doctor. And Lily worked in my lab on a project that was a big interdisciplinary collaborative project across the entire continental US, a project that we affectionately refer to as Stream Pulse. And Lily was really the glue that held the University of Florida's part of that work together. And it really shows Lily's got a bunch of collaborations with people at Duke University and now also internationally to add to the work that she did herself for her dissertation, which is what she's going to talk about today. And I should just add, you know, Lily's came to us as a very accomplished teacher and is teaching middle school and high school science and is I think seeking a career in teaching as well. But she was one of the most impressive graduate students that I've had. She just took a lot of responsibility for things. She was very much a self-starter and just always had a great attitude about working. So we're very sad to see her leave the lab as a student, but very excited that for at least the next several months, she's going to be sticking around to help us out with some projects. So yeah, Lily, it was a great run, and I'm very proud of the work that you did, and thanks for presenting today.
Thank you, Matt. All right, I'm gonna share my screen. Let's see. Before I begin, my son is napping next door. I'm trying to work remotely. So just in case there's an interruption, you know, please forgive me. Good afternoon and welcome. And thank you for attending my seminar. I'm excited to give it. It's about metabolism in our North Central Florida rivers and streams. And I'm going to share some of the things that I studied and learned during my 6 years as a PhD student here. Before I begin, to actually write a dissertation, it takes a lot of help from people all around you. So I would like to thank some folks. First off, my funding sources, the NSF Stream Pulse project that Matt Cohen mentioned, as well as the St. Johns River Water Management District. Their CRIS project funded my research. I'd like to thank the folks at SNRE and SFRC and my awesome committee at UF. And also I had a wonderful lab that not only aided me in studying these streams and rivers, but also joined me in recreating in them. So this presentation is about stream metabolism, and I thought I'd give, for those of you that are less immersed in it, you know, like a fairly general introduction to this corner of ecology. Stream ecologists, ecosystem ecologists, like me, we like to take an entire ecosystem, like this wetland that you see here, and just draw a black box around it. We're not so concerned about individual species inside that ecosystem or populations or communities. We're more interested in a very integrated view of that ecosystem. And when you boil it down, there's two main processes at work in the ecosystem. There's primary production, which is photosynthesis, and then there's respiration, which is what all living things have to go through in order to use the energy that they eat or obtain for work and growth. And so those are the processes that I focus on when I study ecosystem metabolism. And Ecosystem ecologists are also interested in the fluxes of matter and energy that go into and out of the system. For example, one big one is oxygen. All living things would need oxygen, and its counterpart, carbon dioxide, especially in this era of climate change. So that's what metabolism is. Why would we care about stream metabolism? Well, it turns out that these rates of GPP and ER are often unique to ecosystems. And if you put the two together, you can kind of get a metabolic fingerprint, if you will, of the ecosystem. And that can tell you something about the health of the ecosystem. In this example, this is a river in Spain, and what happened is they put in a wastewater treatment plant, and you can see the black was before the wastewater treatment plant, and the green is the fingerprint after the wastewater treatment plant went in. And you can see that shift in the metabolism of the stream. And so it gives that integrated view, just like as human beings, we go in the doctor and they always take our pulse and blood pressure, and that kind of gives an idea of our general sense of health. Metabolism, in a way, can give an idea of the general health of an ecosystem. The other reason to care is, like, in the bigger context of carbon cycling. It turns out that streams and rivers, they get a lot of carbon from the terrestrial landscape, and they also process that carbon, a lot of it, that's the metabolism part, and emit a lot of that carbon back into the atmosphere. So while streams and rivers only make up a few percentage of the continental surface area, they actually process a lot more carbon than their surface area suggests, kind of making them hot spots for carbon dioxide. So that's why to study metabolism. Why study them here in North Central Florida in our subtropical lowland rivers? Well, we have, it turns out, some of the darkest water in the world coming from the Swanee River Basin, as well as some of the clearest water in the world coming from our many first magnitude and smaller springs. And so because the two waters blend into each other as they flow into each other, that's what's pictured here with the springs on the right and the black waters on the left. We have this incredible gradient of dissolved color. And if you're interested in things like how light might affect productivity, which I was, this is like an ideal place to study that kind of light effects on productivity. The other reason to study subtropical lowland rivers is that the tropics and the subtropics are hotspots of CO2 invasion. We are located, at least latitudinally, at that orange line that you see across the graph here. And you can see there's a huge jump in not only the concentration of CO2 in the streams and rivers, but then the fluxes of CO2 coming out of the streams and rivers into the atmosphere. Of course, the tropics, have, even greater amount of CO2 and evade a lot more CO2. But, most studies as far as stream metabolism go happen in the temperate regions and in the boreal regions, and they don't contribute that much globally. So if you care about global carbon cycling and climate change, like, we really need to start paying a little more attention to the subtropics and the tropics. So this is kind of what I see as a roadmap for today's presentation in my dissertation. Obviously, I'm talking about stream metabolism, and there's several drivers of, or maybe many drivers of stream metabolism. I'm going to focus on light driving productivity. And it's in this greater context of global carbon cycling, which is another part of my dissertation. But in order to tie the two together, stream metabolism, is measured with oxygen. Carbon cycling is measured in carbon or carbon dioxide. And so to tie them together, you need something called a respiratory quotient to do it's pretty much a conversion factor. And so that was the third piece of my dissertation, looking at the respiratory quotient, which turns out is almost always assumed and rarely measured, especially in streams and rivers. So the first part. We know light is a major driver of GPP. On the graph here, it's an inner biome study by Mulholland et al. And you have PAR, which is photosynthetically active radiation. So the light that's within the wavelengths that chloroplasts could use for energy, and versus GPP. And we see that the correlation is pretty strong with a pretty high R value. But we also know that not all the light shining down on an ecosystem can be used for photosynthesis. And that difference between the incident light, so the light that's shining down on an ecosystem, and the maximum GPP that the ecosystem can produce is the light use efficiency. Light use efficiency quantifies not only the intrinsic capacity of the chloroplasts themselves to fix solar energy, so how efficient all those chloroplasts in an aquatic plant or an algae are, but it also quantifies, the way it's defined here, the ability of the autotrophs to capture that light and actually use it. And it's often like the rule of thumb is that if you look at light use efficiency using biomass, it'll be 1%. And then if you convert it to light use efficiency using gross primary production, it'll be about 2%. And this is kind of what they've mostly found in terrestrial systems and what's been taught. I think this comes from a middle school science textbook. And I'm sure you've seen that energy pyramid before. And so it's always been 1% of sunlight or 2% of based on gross primary production. But if we look at streams and rivers and we look at the studies where they go, those cross-site studies, it turns out that that slope between light and GPP, and that slope is the light use efficiency, is much higher. Odom here in Florida, in the spring-fed rivers, back in 1957, found that the light use efficiency across springs was 4%. And then if we take the Mulholland inner biome comparison data, and we fit a line to that, it turns out to be even higher at 6%. So what's going on? Are streams and rivers just like super efficient compared to other ecosystems? Or is there something else going on? And that was my focus of this chapter of my dissertation. And so with advancing technology, we're now able to test this light use efficiency idea because you need two things. You need light data and you need GPP data. And I could get both at high resolution by modeling both of them. The light model is based on Julian et al.'s Benthic light availability model, where open sky light is first attenuated by the canopy, and then some of it's reflected, and then it's attenuated by the water column. So if you think about it, the algae and the aquatic macrophytes, the aquatic plants in the streams and rivers that are actually photosynthesizing, they're at the stream bed. So it's really the light that actually reaches the stream bed that matters for photosynthesis. And so I worked on a paper where they were modeling riparian canopy attenuation, and then my main contribution was that water column attenuation of light. And then for the GPP, the advances in our modeling of metabolism allow us to get daily GPP even if we don't have some data that's hard to get, like reaeration data. So that's work done by other people, but I learned how to use all that and get the numbers I needed. So the question for this first part of my dissertation was, will more rigorous characterization of light regimes allow us to better predict maximum GPP, and constrain the light use efficiency. And I thought it would on both accounts. Like I said, my main contribution was this water column light attenuation, and it's an exponential decay curve. And what I did is I went around north central Florida for a few days and went to bodies of water, some dark, some clear, everything in between. Pretty much, like if I drove across a bridge, I stopped and stuck my sensors in the water. And what I was able to do was I did light profiles that gave me the KD, which is the light attenuation coefficient on the y-axis. And then I also, at the same time, put in some sensors that could give me the fluorescent dissolved organic matter, or FDOM. And it could give me an instantaneous reading of the FDOM. And so by doing this, I was able to build this rating curve because we can stick FDOM sensors in a stream and river and leave them there. And then from the FDOM, then I can figure out, okay, based on the FDOM measurement, what was the light attenuation supposed to be? And kind of model the light reaching the bottom of the stream. And I had to do, because as streams flood and dry out, you know, the distribution of depths changes. So I had to do some modeling just to, because this decay coefficient, or this exponential decay, depends on depth. And so kind of model all that. And what I found was that water column attenuation is critical to stream light regimes. So we had in our study sites a gradient of not only color, but stream size. And I tried to illustrate them here for you all. But what I found was even in the, like a really shallow stream, that's, gosh, drain is only maybe sometimes 1 meter wide, 2 meters, you know, 2 or 3 meters at most. And there's trees bordering it on both sides. So it's going to have a lot of riparian shading that's going to block out a lot of light. But even though it was shallow, it's less than 1 meter deep, because it was so dark that the water column shaded almost as much light, or took out, attenuated as much light as the riparian canopy. So water column light attenuation is important. On the flip side, at the Upper Silver River, clearest water in the world, but it's deep. It's about 2.5 meters deep. There's still, even in the clearest water in the world, 43% of the light was attenuated just by the water column. So like my message here was sort of like, we can't ignore water column attenuation of light, even if you have clear water. So I found that benthic light regimes best predict GPP. Here we have just the association between open sky light and GPP. It's not great, you know. There's definitely an association, but it's not great. You know. It gets a little better with stream surface light versus GPP. And then improves dramatically, at least the R value does, if we look at benthic light reaching the stream bed versus GPP. However, if you look more closely, the data from all the different sites have a different symbol. And if you look at each individual site, if you regress that line to get a light use efficiency, it is much shallower than the overall global association regression between benthic light and GPP, which is that black line. Okay. So there's definitely, at least in this model, statistical model, there's a significant side effect here. And so it's like, I have to account for this side effect. It's, now what is it? Well, I think that side effect, because it seems to adjust the intercepts of these different lines. Like the slopes of the lines are very similar to each other. That was the other thing that like really popped out at me. But the intercepts are very different. I think that intercept or that side effect is really the autotroph biomass. If you think about it, if you have this spring-fed river that's got where the stream bed is completely covered with this thick eelgrass, you know, and it's green everywhere. And even if you have a little bit of light, the amount of photosynthesis that can happen in that ecosystem is going to be pretty large, pretty high. Versus if you've got a stream bed, and this is not, I didn't have a picture, but this is not Florida. This came from somewhere else. But if you have a stream bed where there's only like a little bit of algae, or maybe the algae is very patchy, if you have a little bit of light, you barely have any GPP. Okay, so I think that difference between these lines here is that autotroph biomass, which in turn is like a measure of how good that autotroph community is at capturing the light that's available to that ecosystem. Okay. Of course, autotroph biomass is hard to measure. You would have to go out and take samples and dry them and burn them, and it's data that's hard to come by. But a good proxy for that autotroph biomass would be mean light. I mean, if you think about it, if you're in a place where that overall throughout the year gets a good bit of light, there's probably going to be a lot of autotrophs, a lot of algae and plants that grow in that place, versus a place that's like more shaded and doesn't get much light, then you won't have much, right? And so what I ended up doing was I built my statistical models around this idea of benthic light regimes, not just daily light, but daily light and mean light. And when I did that, the correlations got a lot tighter. The regressions got to like R squared of 0.8, R squared of 0.8. And also the light use efficiencies kind of converged on a value that was around 1.9%. So light use efficiencies converge with benthic light regimes in two ways. The first is that you reduce variation in light use efficiencies by thinking about how much autotroph biomass, or as the proxy, how much mean light that site is getting. And then the other thing is, when we do that, our numbers for streams and rivers, they line up a lot better with terrestrial systems and numbers from other aquatic systems. And let's see. Oop. Sorry about that. I was trying to show. I don't know if you can see my cursor, but on the far right of this plot is actually that 1957 Odom study, where they had light use efficiency that was much higher than anything found in terrestrial systems. But when I accounted for mean light and benthic light, or mean light and daily benthic light, then my data, which is highlighted in yellow, kind of falls more in the middle of all these different systems. All right, so it's kind of suggesting that maybe there is a universal light use efficiency across ecosystems. And if that is the case, then it's great news, because we can take that number, and we know we might be a little off, but not that far off. We can take that universal light use efficiency number, and then if we have incident light data, we can make some predictions of what maximum GPP should be in those systems. And this is something that terrestrial ecologists have been doing for a while now, but we can do that for aquatic systems as well, which is pretty exciting. All right, part 2 of my dissertation was centered around this respiratory quotient idea. And again, we need it because metabolism is measured in oxygen, but often we're interested in carbon fluxes, so we need some conversion factor. And the respiratory quotient, or RQ, is basically the moles of CO2 produced divided by the moles of oxygen consumed. And as an example, in a Nature Geoscience paper, Hotchkiss et al. Found that 28% of CO2 evasion came from metabolism. But depending on the RQ that's assumed here, that number could be higher or lower, and significantly so. And so we know that respiratory quotient depends on the organic matter that's being respired. And it's often, the RQ is often assumed to be 1. And that's because if you take, a mole of glucose, you need a mole of oxygen, and you would produce a mole of carbon dioxide. So 1 divided by 1 is 1. But there's some variation depending on the type of organic matter respired. And in freshwaters, the general trend, and this data comes mostly from lakes, seems to be that if you have more labile organic matter, the RQ would be lower. And then vice versa, if you have more recalcitrant organic matter being consumed during respiration, the RQ would be a little higher. Community RQ or ecosystem scale RQ also depends on some other processes, and that might affect it as well. And so, and then just in the literature, if you find a metabolism study, RQ is almost always assumed, and it's very rarely measured. And we're talking just a handful of measurements for every kind of aquatic system. Lake measurements, so other freshwater measurements, they seem to vary between 0.6 and 1.5, which is pretty big variation. And then riverine RQ measurements are very few. And part of the reason is that it's hard to measure RQ. This is a study by Robert Naiman, and he's a pretty well-known stream ecologist. And he found values ranging from -7 to 2.6. I'm not sure what -7 means, but that's a huge range. And so he writes here in the paper, our attempts to measure the photosynthetic quotient, PQ and RQ, will not be presented here because of these facts, although the problems we encountered are worthy of notation. And so it's just difficult to do. And I found that as well, but I tried. So one of the questions of this part of my dissertation was that, what is the community RQ in a highly productive river? I actually wanted to make some measurements. And I thought, I did my study at the Ichetucknee, and because I figured the organic carbon in the Ichetucknee was pretty labile, that RQ values would be very near 1. And I did this part with benthic chambers. So if you were floated down the Ichetucknee like in late 2018 or early 2019, you probably would have seen these monstrosities in the river. They weren't pretty to look at, but they were pretty cool in design. Basically, they allowed me to capture a slice of the water column. So within the benthic chamber, they sunk into the sediment. So I had about 6 inches of sediment. I had the water column, and then they were also open to the atmosphere. So they had reaeration just like the natural river did. They had a pump to mimic the flow of the river, and they had portholes where I could flush the chambers out. But by just like capturing like one slice of the river, I could actually add things to these benthic chambers, to the water, and drive concentrations maybe above or below ambient. And my chambers had oxygen sensors, a CO2 sensor. They were also shaded, and I blocked the light all around it. Because I wanted to focus on the respiratory quotient, and so I had to turn off photosynthesis somehow. And so for half the chambers, I dosed them with sugar, a lot of sugar water, because sugar is very palatable to microbes. They would probably consume that sugar first before anything else. And then the other thing about sugar, glucose is in this case sucrose, is we know the respiratory quotient should be 1. So it was like a good check on my measurements that I got. And then half the chambers were my control chambers where the microbes were consuming natural organic matter. And what I found was that there wasn't any significant difference between my control chambers and my sugar chambers. So the inference there is that, okay, naturally the microbes in the sediment and the water column of this river are consuming organic matter that's very similar in composition to sugars and carbohydrates. But I put a dotted orange line across the graph at where RQ should be 1. And in the sugar chambers, they definitely should have been 1. And they're not. And same with the control chambers. So that's kind of indicating that there's some other processes at work that's repressing the community RQ values. And 2 that I explored were the incomplete oxidation of organic matter to carbon dioxide. So for every mole of oxygen, we're not really measuring, all of a complete conversion into carbon dioxide. So the RQ would be lower. And then carbonate buffering, this is a spring-fed river, it has high alkalinity, and that would affect the CO2 concentrations in the water. And so that would affect RQ. And I'm always interested in how my system compares to other systems. So this is kind of like a literature review of RQ values and in all aquatic systems. And as you can see, there's not many. In orange, I have in the orange little rectangles are the system means. And you can see that in marine systems, RQ is typically just below 1. And then in lakes, they seem to be much higher than 1. And then in rivers and streams, like I said, there's my study, and then there's a study from 1963. They're low. RQ values are much lower. And so, of course, I think there's a lot more work that can be done with this. I think we need probably, as aquatic ecologists, need to take some more measurements. Where I would have, if I had more time, I guess, and funding, I would have liked to design some chambers that could have been used in blackwater rivers, because that organic matter is not at all labile. It's very recalcitrant. It's been chewed over, leftover organic matter from from wetlands, and it would have been interesting to see what the respiratory quotient would have been there, and then between a spring-fed river and the blackwater river, I think we would have the 2 bounds of what a respiratory quotient would be in a stream and river. And so, assuming a thought experiment that the respiratory quotient that I found, respiratory quotient of around 0.8, was universal to streams and rivers, then the number that I quoted earlier, where of 28% of carbon dioxide degassing from streams and rivers coming from metabolism, would have changed to about 19%. So it does make a big difference. And so this is where this commonly assumed number, it seems so trivial, but would have grave consequences for carbon fluxes. And finally, where does stream metabolism sit in that global carbon cycling? Aldo Leopold's daughter once said that rivers are the gutters down which flow the ruins of continents. And one of those ruins of continents is carbon. The terrestrial systems, they pull a lot of carbon from the atmosphere and sequester it and export it. And it fuels a lot of the metabolism in streams and rivers. And so, but if we look at the terrestrial ecologists' mass balance accounting of carbon, and then we look at the aquatic ecologists, the freshwater ecologists' accounting of carbon, they don't match. And here's what I mean. I'll give you some numbers. So in green, this is the terrestrial ecologists have found that globally, 4.5 petagrams of carbon per year is fixed. Is not that's the net ecosystem production of the terrestrial ecosystems. About 2.6 petagrams of carbon per year are stored in soils, which leaves 1.9 petagrams of carbon per year to be exported to inland waters. From the freshwater ecologists, if we use this active pipe model, where we look at all the fates of carbon in a stream and river, So the 3.9 petagrams of carbon emitted to the atmosphere, plus the 0.6 that's buried in sediments, plus the 0.9 that's exported by rivers to our oceans. If we sum all that up, that has to equal the amount of carbon coming into the system, right? But if you sum all that up, that's 5.1 petagrams of carbon. So the terrestrial ecologists are saying 1.9, and the freshwater ecologists are saying 5.1. So who's right? You know, this is a pretty big difference in numbers, right? At the same time, recently, based on work done in the mostly in the tropics, Abril and Borges kind of put out this idea that maybe aquatic ecologists, like we have to put wetlands in our mental model of the active pipe. And the reason is that riparian wetlands, and there's a lot of riparian wetlands in the tropics, they are very productive systems, high net primary production, right? But very slow gas exchange. So a lot of that carbon is now trapped, and these are flooded lands, right? They're now trapped in the water. And because riparian wetlands border rivers and streams and are very hydrologically connected to rivers and streams, a lot of that carbon is exported into rivers and streams. So riparian wetlands are important. And so if you think about the water that rains down in the uplands and then percolates and then slowly towards the river and stream, they have to go through the river corridor, which is that riparian wetland zone, right? And they could pick up all that CO2 that's being, there's a lot of respiration in riparian soils, and they pick up all that CO2 and basically carry it into the stream and river. And then there's a lot of, potentially a lot of processing within the stream and river. And so that's why the stream and river emits a ton of CO2 into the atmosphere. So that's kind of my conceptual model for this part of my dissertation. So far, by measuring the CO2 evasion into the atmosphere from streams and rivers, and then by measuring metabolism, most studies so far have taken the difference and just attributed it to external sources of CO2. And usually by external sources, they mean terrestrial and terrestrial upland sources of CO2. My goal was to partition those external CO2 inputs into terrestrial and riparian inputs and actually see where the CO2 is coming from. And I hypothesized that the mass of lateral CO2 inputs into rivers and streams would be then dominated by the river corridor, which is the stream channel and the riparian areas. And I conducted this study in the Santa Fe River Basin. This is in our backyard, and I had a gradient of stream size, but also very contrasting catchment geologies. The upper basin, which is on to the right on this in this figure, it's got that hawthorn clay layer that keeps the aquifer, the Floridian aquifer, confined, and it doesn't interact with surface waters. And because of that clay layer, a lot of water is trapped on the surface of the land, of the landscape. And so it's got a lot of riparian wetlands, as you can see. The lower Santa Fe River Basin, the clay layer is no longer there, and so the aquifer is unconfined. It's very connected with the streams and rivers, and that's where all our springs along the Santa Fe are, in the Ichetucknee River. There's very little riparian wetland, you know, as far based on catchment area in that part of it. And so I measured the lateral CO2 inputs into the streams and rivers using a very basic discharge times concentration equation. And I found, I found the mass flux for CO2 from the uplands as well as the riparian. So I measured CO2 concentrations in the uplands as well as the riparian zones. along a transect to the stream. And what I found was that these fluxes of CO2 that go into and out of the system, they vary over time, which is the top graph. They also vary over space, which is that bottom graph. And just the way I did the calculations, I didn't feel comfortable making too many conclusions about temporal variation, but I think I could confidently say a lot about spatial variation. And one thing I found was that metabolism, so net ecosystem production, those internal sources of CO2, provided pretty much as much CO2 as lateral inputs into the system. But this is a lot higher than they found in other studies in other regions of the world. Right? I found that stream size seems to control the magnitude of external CO2. So as the streams get larger and larger, the external CO2 proportion gets smaller and smaller. And then I also found that geology plays a role in how we partition between terrestrial, upland, and riparian sources of CO2. So in the upper basin, where the aquifer is confined, it's mostly the riparian sources contribute most of the CO2 that ends up in streams and rivers from outside the stream and river channel. Whereas in the lower Santa Fe River, it's mostly the terrestrial uplands that contribute most of the CO2 into the channel. And then if we take an average across all the sites, I found that the riparian yield, so for every square meter of a riparian wetland, it would contribute 65 times more CO2 than every square meter of the terrestrial uplands. So seems like most of the CO2 is coming from the river corridor, from the riparian zone, or from the stream channel itself. Okay. If these results were general, so pretend like across the world that these results, happen in all streams and rivers. What that would mean, and I did some calculations with those global carbon numbers, that the terrestrial to aquatic carbon transfer would be more like 2.37 petagrams of carbon per year. And remember, the terrestrial ecologists thought it was about 1.9 petagrams of carbon per year. So this number is much closer. So if we're in our conceptual model, if we're thinking about riparian wetlands as well, this number is much closer to the terrestrial ecologist's mass balance. And furthermore, not only does most of the CO2 come from the river corridor, it seems like most carbon comes from the river corridor, organic carbon as well. And remember, organic carbon fuels a lot of the metabolism that happens inside the stream and river. So on the left, we have results from a study in boreal forests, so another kind of system, and they found that the hill slope CO2 coming from uplands is basically, or not CO2, the hill slope DOC, dissolved organic carbon, was basically none. It was all coming from the riparian zone. On the right, we have some results from also the southeastern coastal plains, so something very similar to our systems in Florida. And as you can see, it's the wetland forest, the riparian forest, that is contributing most of the organic carbon to the system. So to summarize, I want to leave you with 3 takeaways. One is that a constrained light use efficiency would allow us to estimate maximum GPP if we have benthic light data. That measured respiratory quotients can vary greatly from the commonly assumed 1. And by extension, this could really misrepresent carbon fluxes. And that the river corridor disproportionately sources the carbon that is eventually degassed from streams and rivers. So that's one way to remember it. Matt Cohen always has his PhD students write a haiku summarizing their dissertation. So another way to look at it is this. Thank you so much.
Thank you so much, Lily. That was awesome. And I love that you ended on a haiku. Never seen that done. So we definitely have some time for questions. If you have a question and you are in the Zoom meeting, you are welcome to either type it in the chat box or unmute your mic. And if you are watching on YouTube, you are welcome to type it in the chat box on YouTube and I can read it to Lily from there. All right, well, I have a question for you.
Sure.
I'm just curious, it seems like you did a lot for this research, and I'm curious what you feel was the most important part of what you did, especially because it seems like you have a really kind of deep understanding on the gaps in the literature and where your study fits in those gaps. So if you could talk about that at all, just what do you think was the most impactful part of your research?
Oh, there are, for the stream pulse project that I was working on, that the national project Matt was talking about, I think that first part of that light use efficiency part, it was kind of like, I would say that for the those particular researchers, I think that seemed to be what they were most interested in, and and I tried to layer my dissertation, and so have many scales, and I think I liked that the third part where it's like, where does stream metabolism fit in the global carbon cycling? And maybe it's just the way my mind works. It's always like, where do I fit in the bigger picture? So I don't think I had I had so many sensor failures. So the data, I just, you always at the end of a dissertation or any research project, you're like, oh, I know how I could have improved it, and I wish I could go back in time. And but yeah, I just kind of wish I had like better data for that third part. Like the data is good, but it's just like I had so many gaps, like it was so disappointing at the end. I think I had enough to like make inferences, but it's just like I think I could have said more if I had better data. So.
Are you planning on building on this research?
I would love to. I would have to find work that would allow me to. Yeah. So
awesome.
That's the challenge. Yeah.
Are there any other questions out there? All right. Well, I think we'll wrap it up then. Thank you so much for taking the time to present to us today, Lily. It was an awesome presentation.
No problem. I was glad to do it. Thank you all for tuning in.
Have a good day, everyone.
Okay. Bye-bye.